I'm Claire McKellar. I'm the Director of Operations and Community for FlyWire, and my fellow presenters today are Mala Murthy, one of the two PIs on FlyWire, the other one being Sebastian Sung, who can't join us today, and also Sven Dorkenwald, who is a PhD student in the Sung Lab. And I would like to mention that in order to post questions, they should be posted into the chat to me, Claire McKellar, the moderator, and I will collect all questions and then I will read them out probably at the end, although we can, if there's anything critical, we can also do it inside the presentation. And all right, I'm going to hand it off now.
Okay, thanks, Claire. So hi, everyone. Thanks for coming. This is our second FlyWire Town Hall, and the purpose today is to provide updates on the state of the connectome, our efforts to complete proofreading and cell identification within FlyWire, the development of tools for connectome analysis appearing in FlyWire, and our plans for releasing the data. And so we feel we've made a tremendous amount of progress since the last town hall, so we're excited to share these updates with you. And I think the presentation will be about 30 minutes or so, leaving plenty of time for questions. So I wanted to start with just a bit of history. Our FlyWire project began in 2018 when Dobby Bach and his team at Janelia released FAF-B, the female adult full brain EM dataset. And that same year, Sebastian Sung and I applied for and received funding from the Brain Initiative to support realigning this data set, segmenting it, building a proofreading system that we call FlyWire, and disseminating the system widely to the Drosophila neuroscience community. And I'm happy to let you know that Sebastian and I recently received 2 new Brain Initiative grants to support the future of FlyWire over the next 5 years. So Sven, can you move to the next slide? Thanks. So the early dissemination served 2 purposes, really. By openly releasing the data as it was being proofread, together with automated synapse detection from Jan Funke's lab, we enabled a large community of neuroscientists to gain information about their circuits of interest. And this has already resulted in a number of publications using the data, which you can see here, and more are forthcoming. Sven, can you move to the next slide? Okay, so these efforts, together with the work of full-time paid proofreaders at Princeton and with the help of proofreaders from the Cambridge Connectomics group, these efforts are collectively building what is poised to become really the largest whole brain connectome to date and the first for the fly. And so as of today, we've completed over 1.2 million edits of the data. So each point here is an edit within the data set. And these include, as you know, splits and mergers of the raw segmentation. And Sven Dorkenwald, who's a Princeton computer science graduate student and the leader of the FlyWire project, he'll provide more details on this effort momentarily. But the majority of these edits have been in the central brain, as you can see, but we're also making progress in the optic lobes. Okay, can you move to the next slide, Sven? So our FlyWire member labs, as you know, come from a large number of universities and research institutes worldwide. And these numbers are steadily growing. Claire is continually training new users to join FlyWire. And I just want to mention that as someone who's worked on Drosophila neurobiology for over 20 years, I've always known the power of this community, but it is certainly gratifying to see how member labs, from diverse regions, many who have never worked together before, have come together to collaborate on what has really become an incredible resource for the field. So I am extremely happy about this. And Sven, could you go to the next slide? So before I hand things off to Sven and Claire, I would like to thank our FlyWire Advisory Board, who provides oversight on the resource and assists with community issues that arise. And ours is certainly an experiment in open science, and we lean heavily on these individuals to make sure that our principles and policies are fair and equitable. So thank you to all of them. And with that, I would like to pass it off to Sven, who will give you an update on what's going on with proofreading and cell identification.
Yes, thank you, Mala. Hello, everyone. as Mara just mentioned, we have passed about 1.2 million edits by all of you, and that is about twice what we had at the time of the last town hall. So a lot has been going on, and even the number of users has been increasing. So with the next slides, I will give you an idea of what that means in terms of completion and to what extent the dataset has been proofread, and what is coming next and what we expect in terms of timeline. The way how you can see this kind of data is through NeuroGlancer. So several weeks back, we introduced colors for our light bulbs here on the right. And after the last town hall, we asked all of you to submit completion data for all these neurons, which we have uploaded and made available, and also continuously add to it by adding more features to this light bulb where everybody can submit completion information about any neuron that has been proofread. So here on the right, you can see that neurons that have been marked as proofread show a green light bulb. That means this version of the neuron or previous version has been marked as proofread, whereas those that have not been proofread, like this one here in the Optic Globe, is shown with a green with a yellow light bulb. Behind this, we built a service which we originally called Proofreading Management, but which is really growing to be a service that hosts all kinds of data that is also including cell identification. So usually when you click on these buttons in the light bulb, you will be forwarded to here. And here we're also building up more tools that will also allow you to search, for instance, cells by their identification. But at the same time, we're also trying to integrate the service as much as possible into NeuroGlancer, so you can do as much as possible and through NeuroGlancer and see this information readily in there, which is very valuable, of course. At the last town hall, we set 2 milestones ahead for proofreading. The first one being the central brain itself, which we estimate to be about 48,000 nuclei by a rough estimate based on spatial location of nuclei, which is, of course, not highly precise, and then a second milestone for the whole brain. For the next slides, I will only focus on proofreading within the central brain. And we changed our inclusion criteria for neurons that we think should belong to the central brain. And we now define this as how many synapses does a neuron have within the central brain, meaning the synapse being within a brain region, a neuropil that is associated with the central brain. And this cutoff will end up somewhere between 10 and 100, and that will also depend to some degree on to how small of a fragment we will be able to proofread in the end, actually. Also at the last town hall, we announced that there will be 2 steps to this proofreading effort. So the neuron-centric proofreading, which is the most common one, and I also believe that most of you have been doing, finding a neuron of interest and then proofreading it to a degree that is good enough for analysis. However, this will necessarily miss some neurons, also because some neurons don't have a nucleus in the data set. That makes them hard to actually find automatically. So there is a second step necessary. That's also similar to how the hemibrain was done, which is synapse-centric proofreading or orphan linking, meaning stepping through leftover fragments, going down by size, number of synapses, for instance, and then proofreading them. So we are happy to announce that the first step is basically completed, and that is thanks to all of your submissions, and also some additional effort at Princeton and Cambridge, the Jeffries lab there, to fill the gaps to proofread terra incognita that nobody has proofread. So at this point, when you browse NeuroGlancer and you click onto neurons in the central brain, you will most often see that the light bulb is actually turning green, and that's because of that. So analyzing this data, we have about 54,000 unique neurons that were marked as proofread, and amazingly, this was done by 95 different users. And now we can ask the question, how many synapses have been attached to these neurons that have been marked as proofread? And this is a method of evaluating this data that is very similar to how it was done in the hemibrain. And this gives us an idea for how successful this proofreading has been. And this is how this data looks like. We can ask this question for presynaptic and for postsynaptic segments, synapse sites. And currently in the central brain, we are ranging at about 85% of presynaptic locations being attached. That is the red. I'm sorry, there's not a legend right now. So 85% of presynaptic sites have been attached. A little bit more than 30% of the postsynaptic site has been attached in the central brain. The big discrepancies, of course, because twigs are much harder to proofread. And also as a comparison, this number of postsynaptic accuracy is quite close to what has been reported in the hemibrain paper. Of course, we can do this for all brain regions, and I will post an update of this here again on Slack later for you to look at in more detail. But as a summary, we can also look at a histogram of all the brain regions, and we can see here that most of them are indeed over 80% accurate on the presynaptic side. And on the postsynaptic side, there is actually quite a spread for the brain regions in the central brain. And that is just because most likely the prevalence of twigs is different between brain regions. Now, zooming in on these proofread neurons, we have about 42,000 neurons that have over 100 synapses in the central brain and that has been marked as proofread. Here I'm showing you a histogram of all of these by the number of synapses. There is, of course, a small tail, and this is because there are some neurons that go into the central brain, but are actually mostly in the optic globe. And additionally to those 42,000, even though it was not a focus, but of course, many of you are interested in it, there are about 12,000 neurons that are primarily in the optic lobes. And we can also plot these now by their number of pre- and postsynaptic synapses. And we can see that they are highly correlated, but of course, we have usually more presynapses per neuron than postsynapses. And again, that is because twigs are much harder to attach. And I'm also showing this just to make you aware of this again. Now, the step ahead is the Synapsancy proofreading. And we have to do that because there are fragments that have been missed, but also because they are just neurons that don't have a nucleus in the central brain. And those fall into categories of ascending neurons, sensory neurons, and of course, the missed nuclei. This effort will be done mostly or we imagine this to be done mostly at Princeton and at Cambridge again, also because this is an effort that is not close to many research questions necessarily, and it's just going down a list. So this is something that we will provide to basically finish or try to complete the resource as a whole. But in a second, I will also mention another step, which is review, where we want to include, again, the whole community here. To give you an idea for what the road ahead is like for this step, there are about 23,000 segments that are unproofread that have more than 100 synapses. With our current speed, I estimate that we will take for this about 7 to 8 weeks. Though even throughout this process, even within the next weeks, we will most likely capture all the fragments that are actual neurons. And after that, it will more and more be like only fragments that will be attached and not whole new neurons added. So the resource will get more and more accurate, but most neurons will already be in a database of proofread neurons. There will be, of course, a long tail ahead for us where we have to check for outliers. Neurons that are very small, for instance, will be a challenge to find. But I believe that in a few weeks' time, the vast majority of neurons will be in the database and good enough for analysis. And then, of course, we need to evaluate the resource, and we also want to allow anybody to submit review of any neuron that has been marked as proofread, because there are still mistakes happen, and that is totally human. So we want to be able for anybody to flag such neurons as good or having some kind of issue, and we will be able to use that to drive proofreading and also to build up an evaluation of all these neurons that we can then use to evaluate the resource. And again, this will partly be coordinated, but also anybody of you will be able and will be asked to submit some of those information. So yeah, this is something to look out for. We hope to release that quite soon. It will come through the light bulb as all the tools that we had so far coming out of NeuroGlancer. So we will be able to not only submit completion information, but also review. And then as we continue finishing this resource, a larger challenge that is coming up, and some of you have mentioned that to us, is that once you feel like a neuron is finished, but somebody else is still making edits to it, people would like to preserve the proofreading that is already there. There is a trade-off because, of course, somebody may find a small error and wants to edit that, so we will never lock proofreading of neurons entirely. However, what we are planning on doing is we want to add friction to the process, such that once a neuron has been marked as proofread and then even evaluated as good, we will add prompts and other kinds of frictions that will make it harder, I mean, even restricted people that can make edits eventually to have the resource converge and reduce proofreading of the neurons that are in the proofreading database. And then finally, this was all about a central brain. The Optic Globe is, of course, still out there as well. We now added, and this will be released later today, a way to also proofread there and just check out cells. This will be basically neuron-centric proofreading all over again. We can just check out cells to proofread in the Optic Globes and contribute to that in the long run. And this will be a combination again of just proofreading cells from scratch and marking them as complete through the light bulb. So as a summary, the central brain is coming in on completion. Of course, it will never be perfect, but on something that we will deem good for analysis. And something to look out for there is a review system that allows anybody to submit to judge those neurons and flag problems. And the Optic Globes will now basically also start an effort that will eventually lead to a whole brain connectome. And as you can see here, of course, completion there is much lower, but that's our eventual goal, of course, to make the resource of the whole brain. Good. With that, thank you so much, and I will pass it on to Claire.
Thanks, Sven. Could you go forward a slide for me, please? So Sven was just talking about proofreading, but the other really important aspect of an upcoming connectome will be cell identification or names for as many things as we can find. And so there's been a huge community effort to enter cell names as well as to do proofreading. And I'm happy to say that now using that light bulb, you can now click a cell to see any name information that people have entered. So far, 23,000 cells in the brain have some kind of identifying info, although that can vary in level of detail. Someone might have just put in, that this is a descending neuron, and they might not have yet had time to figure out exactly which descending neuron it is. Although that's actually probably a poor example, because I know that the people working on descending neurons are putting a lot of effort into correct naming. And I'm going to announce that coming soon we'll have text-based searches, but that's not yet available. So right now you can, if you can find a neuron in the brain, you can see what name it has associated with it, but we will soon let you type in different names to try searching on those. But you can already do that kind of searching programmatically. We would like to encourage everyone to sign up to help contribute names. If anything, that's more critical now than helping with proofreading. And you guys are the experts. Everyone knows so much about their different brain areas that they work on, even cells that you recognize from the literature that you haven't worked on yourself. So here are some of the people who've signed up so far and pledged to help with these cell types. It is fine to overlap with others. We encourage very active coordination to help prevent misunderstandings and make sure that we are all working together successfully. To find out how to join the cell identification drive, go to our homepage, flywire.ai, and then click on tools, and you should be able to find your way from there. Next slide, please. So how do you find a cell? Let's say it's one that you know about. Let's say you've got a confocal stack of it because it's one that you've discovered, or maybe it's one that's already known in the literature and it exists in another data set like the HemiBrain or Fly Circuit or virtual Flybrain. We now have a tool called the FlyWire Gateway with thanks to Ari and Sven in the Murthy and Sung labs that will help you take any one of these neurons and create a point cloud, and then you can use that to click cells near that point cloud within FlyWire to try to find a match. However, a much more sophisticated morphology-based searching is also available programmatically with thanks to tools from the Jeffress lab. And we will try to make those available to non-programmers in the future as well. But this method of point cloud and clicking is actually remarkably successful. So I encourage people to look for any cells they think they can identify. And it doesn't even have to be from any of these sources. As I say, if you know about it just from a paper, then you can use the information that you can get to from these same links to find particular brain areas and orient yourself that way. And frequently you can find a cell that you know about, even if you just know about it from a two-dimensional figure in a paper. Next slide, please. So how to name cells. Our system is a bit different than many others. So all information is accepted, and we're kind of going for the crowd wisdom in the same way that proofreading is crowdsourcing to all of you. And so we are allowing different degrees of certainty. And so one example is that people might add increasing or sometimes even conflicting details. So the first person might say that they think that this cell that they found is a descending neuron. Another person might say, yep, but it also looks like it could be the giant fiber, question mark, with the word putative in there. And then person 3 might be totally certain that it's the giant fiber. And so they put in alternate spellings for it. They put in its abbreviations. They put in a paper in which it was discovered. And this last thing that you see in this list is a number, and we are going to talk about what that number is in just a moment. If a cell is not known in the literature and your lab is planning a naming system for an upcoming paper, please enter explanatory information. So you might say, Smith Lab Neuron 2003, we are working on identifying all these neurons within this lineage that we think is involved in courtship or whatever you want. So the more information that you can provide, the better. Next slide, please. So I would now like to introduce Claire Pilgrim from the virtual Flybrain. She's going to give you more of an introduction to what VFB is and how the naming system that it uses is a great way of getting a very sort of regimented and helpful set of names for neurons. So I am going to now unmute you, Claire, and ask you to share your screen if you can. 1 second, okay. Unmuting you. Thank you. Go ahead.
Okay, can you see the screen okay?
Yeah.
Yeah, great. Okay, hi, I'm Claire Pilgrim. I'm the ontologist for Virtual Flybrain. So VFB integrates neuron data from multiple sources, including EM and light level images from large data sets such as the HemiBrain, as well as smaller submissions from individual labs. And we annotate individual neurons, such as the example below from the hemibrain dataset with cell type terms from the Drosophila anatomy ontology. And in this example, this neuron is annotated as an adult lateral horn AD1A1 neuron, which is a term from the anatomy ontology. So what are some of the advantages of using ontology terms? So you get a persistent identifier to uniquely identify a cell type, and the focus is on the term meaning rather than the label. So each term has a definition based on reference publications. And if you look on the left, this example is for MBON1, and there's a description here. And that description is based on several references, which are at the bottom of the information for this term. And because the focus is on the meaning, the actual label is less important. Each term has a unique label, but there's also a collection of synonyms. And this one also has synonyms of MB, M6, and MBON, gamma 5, beta prime 2A. And all of these can be searched to find the same term. And the ontology is hierarchical. So you have these specific terms for MBON1, MBON2, etc. But they're also grouped by a general MBON term. And all of the MBONs fall under adult neuron with lots of other cell types as well. So you can choose a term that fits your certainty level about what you're annotating. And you can also pull out neurons annotated with terms at different levels. So you might be interested in getting all of the neurons that are MBONs, and you can do that if they're annotated with ontology terms. And this also allows finding neurons of the same type across multiple data sets. So you might have a cell type that's called something different in the hemibrain to what it's called in FAFB, say. But if you have both of those cells annotated with the same ontology term, you'll be able to find both of those based on that term, even if they're called different things in their original data sets. So how can you find a cell type term to annotate your neuron? You can find these terms using the VFB website. So this is an example of a search. You can filter the search if you click on this, on the little 3-line arrow thing in the search box. And in this case, we're looking at adult neurons. So if you can click the check boxes for adult and neuron. And you also don't want to be looking at images because we're interested in ontology terms. So if you click twice on the box next to the image, that will remove any images from your search. And here we've searched for MBON1. This is a synonym for the term that we want, which is mushroom body output neuron 1. So it will find that. And if you click on the top result, that will take us to the term info page for this ontology term, this cell type. So as I mentioned before, each cell type in the Drosophila anatomy ontology has a unique and persistent identifier. And these all have the format shown here, beginning with FBBT. And I would strongly advocate annotating with these identifiers because they're persistent. So it's possible for like names and stuff to change, but the identifiers will persist. However, these are not easily understandable by humans without looking up the label or definition. So it might also be useful to have some kind of human readable annotation as well. So we also have labels for our cell types. These are unique in the ontology, but it's possible for them to change. So I wouldn't recommend using these as primary annotations, but you can also use these in your annotations if you're allowing multiple annotations on each neuron. Many cell types in the anatomy ontology now have a neuron symbol. Most of these are based on hemibrain cell types, and these are also unique, but they're shorter and probably a bit more convenient than the term labels. They're also expected to be a bit more persistent than term labels. However, it is still possible for them to change, and not all cell types have a symbol yet. So these are also probably not great for primary annotations, But if you did get the FBBT identifiers, so the numbers into FlyWire, it could be possible to pull in the labels and the symbols as a human readable secondary annotation. It would also be possible to use your own annotations if you prefer that as well. But once you've got the ID in, it doesn't really matter what else you call it, because the ID specifies the concept of that cell type. So as an alternative to manually linking your neurons to cell types from the anatomy ontology, it could also be possible to use existing annotations on the same or similar neurons. So many FAFB neurons from CatMade and hemibrain neurons are already annotated with ontology terms, and we have these annotations in VFB. And if you can identify the same or similar neurons in FlyWire, then these FlyWire neurons could be typed using existing annotations on the similar neurons. So thank you for your attention, and I'll take any questions to the end.
Thank you, Claire. That was great. So we would like to strongly encourage people to use annotations from VFB wherever possible. But we'd also like to strike a balance, and we don't want anyone to feel intimidated about putting in information. As I say, all information is useful. And if you are not certain about a cell from VFB and you just know something general about it, put in the general thing, and the next person who comes after you might be able to find it elsewhere. So that's great. Okay, Sven, do you mind sharing your screen again, please? Okay, next, I think advance 2 slides. Great. Okay, and back to you then, Sven.
Yeah, this was a minor announcement that we added a new tool that allows you to interactively analyze connectivity data for single neurons. So Jay Geiger worked on this, and this tool allows you to put in a neuron ID and spits out to you the neurotransmitter information. So it queries that table to show you distributions of neurotransmitters pre and post-synaptic, as well as the neuropulse from where these synapses are. And additionally, it shows you the partners ranked by the number of synapses, allows you to also build neuroglandular links out of these that you can then analyze further and find new neurons of interest that you may want to query this way. I also want to give a shout out here to Stefan Gerhardt and his Brain Circuits IO tool, which has some similar properties and allows you to query synapses of neurons as well. Okay, back to you, Claire.
Okay, so when you are ready to publish FlyWire cells, please see the instructions on the website, again, under tools. And Sven, could you go to the next slide and just play a pre-recorded, very short introduction to publishing? If FlyWire has been useful to you for a paper that you're writing, please go to our homepage, flywire.ai, and then to the principles and review those, particularly this short section. Before using cells in a publication, you must first obtain the agreement of every lab that identified those cells, contributed more than 10% of the edits on those reconstructions, or assessed them to be adequately complete. And this applies not only to FlyWire cells themselves, but also if you used FlyWire to guide your tracing in other EM software platforms. So please keep a list of any cells that are that you ever use, and then contact the labs that have contributed to them. And we encourage labs to communicate early in a project. Please share plans, form agreements to collaborate, keep each other informed. Then when you're ready to publish within FlyWire itself, use the light bulb menu, and you can look up who has contributed proofreading edits to a cell using the change log. And you can also, within here, there's a multi-change log that lets you look up many cells at once. you can click on cell completion details to see what users contributed information that the cell was finished. And you can use cell identification to see what users put in name information for that cell. Then email the contributing labs to ask if they agree to your publication. If they don't agree, you can appeal to FlyWire management. And if necessary, you can escalate to the advisory committee, but we will do our best to mediate without escalating. We encourage everybody to be generous in both offering credit and in declining credit to make it easier for everybody to publish. And we think of this as really an endeavor in open science, where many of us might do work that we don't expect direct credit for, but we are contributing to a resource that improves the whole community. Then email us a list of cells you would like to publish, and we will make them publicly available so that reviewers and readers can visit links without needing logins. Include in your paper a list of IDs of the cells used. Give credit to the contributors in the manner that they requested. They might have said, I'm good, I don't need any credit, or they might have asked to be in the acknowledgements, or you guys might have agreed to be co-authors. And finally, from our homepage, we have a section here called credits. And so for any work that Princeton contributed to cells that you are working on, please use this language in your acknowledgements. And finally, please cite the publications that are mentioned on this page. Thank you. Okay, thanks, Sven. Next slide.
Mhm.
Mala, would you like to jump in here and we can discuss this together, and Sven as well? So I'll kick us off and say that thanks to all of you in the community, we are pretty close to having proofread the central brain, and with all of your contributions for identifications, we intend to publish the central brain not earlier than June. And Mala, would you like to make any comments here?
Yeah, so I think we're definitely on target for, as Sven said, completion over the coming weeks. And so we'll certainly keep everyone updated. In addition, we view this as a large scale community effort, as I said earlier. And so the plan is to, of course, be very inclusive with authorship and aligned with the principles that Claire just detailed. We haven't fully finalized the details yet. So, you could imagine there are people who have signed up to use FlyWire, but then in the end didn't contribute proofreading. And so we will have to sort through the final list. But if you have any concerns or questions, feel free to reach out to us and we are happy to discuss with you. We don't have a specific date yet for the central brain data release, but we, as I said, intend to keep you updated as we near that timing.
Sven, do you have anything you want to add there?
Not from my side, but I think that's also the point about combining multiple papers, depending on where your all research is, if you want to publish in conjunction to such a central brain paper.
Yes, that's right. So Claire sent out an email letting you know if you have a paper you're preparing, we're happy to coordinate timing with you. And so many of you have already gotten in touch, which is great. And actually, Claire and I have been having meetings with individual PIs, but some of you we haven't heard from. So if you'd like to meet with us, please do so. And our plan is to coordinate as much as possible with everybody who is planning a paper in the upcoming months.
Yeah, absolutely. Thank you. Sven, next slide.
Yes.
So our largest interest group within FlyWire, I think, are people interested in the subesophageal zone. And we would like to invite all of you and anyone else who's interested to join a 1-hour discussion on April 20th at 11 a.m. Eastern. I myself can count about 80 users working in the SEZ that I know of, and so I will email you guys. But if I've missed you, I will also announce it in the FlyWire forum. here are some pictures, for example, here are a few of the taste inputs within the SEZ, and I've been volunteering to try to identify the motor neurons leaving the SEZ, and there are many labs working on connecting. We want to make sure that we all are aware of what each other is working on and help each other, and maybe come up with a plan for whether we want to address this in upcoming publications. So please join in on that. And I'm going to ask you guys, the people working in the SEZ, to consider presenting at that meeting. A couple of slides, show us what you're working on. I think the more that people are aware of what each other is doing, the more it helps prevent any possible conflicts. So this is just one interest group, but there are many people working in different systems in this brain. So we would like to encourage other interest groups. And if you want to help to organize an interest group meeting, perhaps for the visual system, let's say, or we've got labs working in glia, let's say, feel free to reach out and I will help to find all the people with common interests. And we really, in the same sense that FlyWire is a grassroots organization in many ways. The same thing will apply for coordinating efforts for interest groups and their publications. So please consider that. Thanks, Sven. Next slide.
Yeah, and wrapping up the presentation, of course, I want to acknowledge all the hardworking people between the Eye Institute, Princeton, and Cambridge that make this resource possible and working in the background. And of course, funding, especially from the NIH. But with that,
and all of you guys.
Of course, that is.
Yeah.
With that, I think we can open up.
Okay, so please post your questions in the chat, and I will read them out. So the first question that has come in, and Sven, perhaps you could answer this one, is there are some neurons we submitted as proofread via email, but they're not seen as proofread yet in NeuroGlancer. Should we do it again through NeuroGlancer?
Yes, so if that is the case, please email me. There are a small number of neurons that I had to beat out because locations and IDs did not match. If it's a larger number, I may have missed some, so please reach out about those. In either case, yes, you can also submit them through NeuroGlancer, and please feel free to do so at any time.
Great. Okay, there's another question. Is there a way to mass add neuron names? I have several hundred neurons of the same type, and I would like to add their names.
Yes, so I think I should take that as well.
Please, yeah.
Yeah, so we will add a bulk submission feature to the website that is not there yet, simply because verification of some of the data takes too long for web service. So in the meantime, please submit this by email and I will upload it. At the same time, if you want to, please submit it by email and I will upload it. That's the best way right now.
More questions? If you want to speak to, Claire could also unmute you.
Yeah, that's true.
You can also raise your hand.
I've gotten a long comment here that I'll read out. Spectator, not a contributor, still at the sandbox stage. That's if anyone doesn't know, that's the practice data set. Research interest, mechanosensory projections into adult brain and VNC, which are nuclei not in the data set. I'm interested in using FlyWire for education in undergrad neuroscience courses, lecture and lab, either just for student display and exploration of annotated neurons, intro courses, or for crowdsourced annotation, proofreading in advanced courses with graduated, or a manual on-ramp for educators and students. Any suggestions for pre-annotated neurons for student exploration? Anyone already working on this? These are great questions. I'll take that. So yeah, we are interested in trying to open FlyWire to the public at some point, and this has been working slowly but steadily in the background. And we will hopefully try to gamify the system a little more to make the training not so focused on scientists. And we have a whole team of people under Amy Sterling, who's here with us today, who have worked on the iWire game from the Sung Lab. And so we are thinking about this. And we've already tried some pilot projects. So we've got a high school class in Taiwan who's used FlyWire. We've got an undergrad class at Carleton College in Minnesota who's used it. And another undergrad class under Jinsop Kim in Korea. Actually, that's not an undergrad class. I think that includes graduate students and postdocs. And we've even had a little pilot project for citizen scientists. And so we are working on making it more accessible for non-scientists, and we will continue to try to develop that, and we'll let you guys know how that progresses. Okay, next, Dustin asks if he could read, or if he could participate in it with a question out loud. So Dustin, I am going to unmute you. Give me just a second. Okay, please go ahead.
Hello, can you hear me?
Yeah.
Great. Thank you so much for that town hall. It's really interesting seeing all the progress. So my question is in regards to when we submitted the coordinates for the neurons, you asked that we put the coordinates in the center of the soma. I found that when editing neurons, often the soma tract will change because that seems to be the least accurate part of when we're editing neurons, especially in the Optic Globe. I do a lot of work in the Optic Globe, and it's really easy to get the wrong soma in there. So I was wondering if we could do the coordinates in the backbones instead, because when the cells are complete, that's a lot less likely to be changed.
You should take that, Sven.
Yes, so yes, we have come to the same insight on that point for exactly that reason. And yes, the backbone at this point is probably a better location to place the representative coordinate, and please do so. And yeah, for that reason, there are not many, but a handful of neurons that have been Marcus proofed, but probably cut off their primary neuron, and we are, it's on my list to kind of go through these and clean those up.
Yeah, we can update
Very
instructions to the community soon. Thanks.
Thanks. Can I ask
Okay,
another
next
question too?
Oh,
sorry.
sure. Yeah, go ahead.
I was just wondering if there's a way to programmatically access the proofreading information and identification information, because I have quite a lot of neurons and I don't know if I would want to go through each neuron by hand.
Yes, there is, and I can repost to the Cave client. There's a Python interface that allows you to query all this information and access the data as a pandas data frame. You can even request it filtered by user ID or by other information or bulk, and then you can analyze this data on your side. And I will later in a general channel will post again commands how you can do that.
Great, thank you.
Thanks, Dustin. Okay, now I'm going to unmute Marta Costa.
Thanks, Claire. I had sort of three questions, all related to the cell typing. So one was, I guess you're encouraging people to use the cell type identifiers that Claire from VFB described, so the FBBT identifiers. Did you have a plan of how these particular annotations should be added to the annotation framework as it is? So because Claire sort of noted that you can have the numeric identifier, but that actually is not very human friendly when you're looking at things. And so you might want to have some label. And so are you encouraging people to, I know, submit those together, for example, either?
Please do. Yeah, you can use commas or slashes, or really we're allowing everything, which gives the poor computer science people an absolute fit. But we're really allowing entries of any form. But so I would encourage, if you were doing giant fiber, you could type giant fiber, comma, full name of whatever the official name is in VFB, comma, abbreviation used in VFB, comma, FBBT number, comma, degree of certainty, like I'm absolutely positive that this is the giant fiber, things like that. So yes, put in everything that you can find, if you can find it in VFB. And if you can't, anything you might know about that cell.
Okay, thanks. And could I ask then, on the paper coming out, what are you expecting to release? And I asked that more in relation to the cell typing, but also it'd be good to hear if you're releasing skeletons, for example, or meshes. But on the cell typing, for example, as you mentioned, there are submissions at different level of resolution, right? And so how will those be assessed as contributions? Because I know if someone identifies a particular cell type, that's probably more valuable than someone just saying sort of the general group of the neuron.
Could I field this question? And
please.
Yeah, if you guys want to chime in. But basically, at this stage, we are open to feedback, either to us or to the advisory board. But we see all of those as valuable contributions. But I agree, Marta, that like, the naming of a neuron specifically might be more valuable than a general label, et cetera. And so these are the discussions we're having now, which is why I said that the plan is to be inclusive, but we haven't developed a strict set of rules. So if you have recommendations, we're certainly happy to hear them. But our overall goal is to be as inclusive as possible, because we're very indebted to the community for participating at all levels, and people have contributed in the best way they are able to. Yeah, I don't know if that fully answers your question.
One thing that I would add is that, so Marta's absolutely right that putting in a specific name for a neuron is more valuable than a generic name, but someone might have spent hours finding the cell for which they added a generic name. So someone might say, well, I can find all the gustatory sensory neurons if I go into the nerves coming into the SEZ, and I can click on, axon after axon after axon and try to pull up all the ones that look like taste inputs. And that would have been a lot of work, even if I then didn't go on to say this one is a bitter neuron and this one is the sweet neuron or something. So then the person who comes along and names it as a bitter neuron would have had the advantage that of the person who spent a lot of time clicking around to find all those sensory neurons. So although the name started out as being generic, that was a contribution. So I right now we are considering valuing all contributions.
Yes, so I can see those scenarios. I guess in our case we have done a lot of the work in identifying these neurons.
Yeah,
so it's
yeah.
not the sort of the two groups that are doing things in separate in that case.
That's true. Yeah, yeah.
Can I address the second part?
Yeah, please.
Of Marta's question. So you asked about meshes and skeletons. Meshes are already available, at least programmatically, to download for any neuron at any point in time during proofreading. And this will be made available. And we can also make a bulk download available for these if we should, once we publish the whole central brain. And then skeletons are available. And we are also working on making the process of creating skeletons at any point in time during proofreading very fast. And we have most of that in place. So we're planning to provide a service that allows you to download skeletons basically within seconds. And that is coming, but not available yet. But even right now, we have tools to generate skeletons, and I'm happy to post that too.
Thanks, Sven. Yeah, and I think just to add that, I think it'd be great if people do use the cell type labels, it will make it much easier once the paper is out to actually get it integrated with other data that's already published and available on VFB as well. So that's actually a lot of added value on that integration.
Yeah.
Great. There's a question that I'd like to read out. How far are the plans for adding a searchable option for submitted neuron names within FlyWire? Sven, could you take that one?
Yeah.
Poor Sven, I ask him this about every 2 days for the past year. He knows.
So we have a prototype for searching. So there will be a ranked search solution. Basically, you can type in the name and it will show you the names that are most closest to that, based on what people have, anybody has typed in, and we'll try to do substring matching. So you will see a prototype of this, or a version that might be slow over the next week, and then it will get progressively better and faster. But yeah, that is coming really close to that.
But we do have instructions for how to programmatically search the names that are submitted, and Sven could repost that again in the forum, but actually maybe you already mentioned that, but yeah, just a few lines of code, you could actually look up whatever identifiers you're on the hunt for and find their IDs.
Okay, and then there's one last question, and I think then we'll wrap up. This one comes from Salil. Is there a plan to have a connectivity tool to search for connections beyond direct connections, like the shortest path tool in the HemiBrain?
Yeah, we're actively working on that analysis locally with postdocs and students working at Princeton. And as soon as these things are developed, we're planning to add them as features or tools within FlyWire. So I would say stay tuned. It's all a grassroots effort. It's either members of the community or our own lab members who are working on these things. So they're as they're being developed, we're planning to openly release them. But
and we'd like to encourage everyone. You guys. Yeah.
Yes, I was going to say that if you are developing tools in your labs and you're willing to share them, we will be happy to work with you to get them onto the website and to provide access through NeuroGlancer. So please reach out to us.
And there's already, maybe Stefan Gerhardt would just like to take a very brief minute to remind people of the tools that he has. Stefan, if you're still here, I can unmute you. Let's see. Okay, just unmuted you.
I think you re-muted him accidentally.
Oh, sorry.
Hello,
can
you hear me?
Hi, yeah.
Okay. Yeah, yes, actually, like in the next 2 or 3 weeks, I will release a completely new version of the Brain Circuits IO interface. And I will post that on the Slack channel. And yeah, it will allow a couple of new things to do with tables and querying data as well. So yeah, I'm happy to share that on the Slack channel once it's available.
Great.
Yeah,
and actually, I will take one more question because it looks like Lou had his hand up. So Lou, would you, I can unmute you. 1 second. Okay, go ahead.
Yeah, sure. I was just going to mention that as soon as the data is available publicly, we will download it into NeuroGlancer. And that way people will be able to make the kind of queries that they are accustomed to.
Did you mean Newprint or
Newprint? Yeah, sorry. Yeah, it is in NeuroGlancer already. That's right, Newprint. But
could you explain a little more for people who might not be aware of how?
Well, Newprint already has a bunch of query stuff in it that includes shortest path and query by name and all these other things. Query by type, you can do cipher queries and stuff like that. And so it's a pretty general way to look at a connectome. And so once it's publicly available, we will download the same data into Newprint, so then you can use those features if you wish.
Fantastic. Great. I'll work with you, Lou, to get some links into our list of tools.
Okay. Yeah, and the same,
Great.
I mean, obviously you could do the same too, the other way. That there's nothing to prevent people from uploading the HemiBrain data into your query tools which would give people the best of both worlds in either case.
Yeah, great.
All right. Well, thank you everyone for joining us. This has been recorded and we'll post it. And also Sven is happy to post the slides as well in the forum. But yes, thank you all for coming.
Thank you, everybody. See you all in the forum, and stay tuned for more announcements.